Hello and very good morning to all of you, and welcome you all in the one-day seminar on concept of PAT in pharma industry, organized by Adarsh Shikshan Prasarak Mandal, KP Patil College of Pharmacy, Dharashiv. As a PAT, that is a process analytical technology, is a methodology used to design, analyze, and control manufacturing processes in pharmaceutical industry, and it is an important tool for efficient monitoring of process parameters to produce a high quality and safety finished products. So there is a need to understand and upgrade the knowledge about this said topic. So our college has organized this seminar. First of all, I would like to give my warmest welcome to the guest of honor and the speaker of today's seminar, respected Sachin Bhusari sir, Assistant Professor, UDCT Dr. Babasaheb Ambedkar, Marathwada University, Chhatrapati Sambhajinagar. Welcome, sir.
Thank you,
Next,
madam.
I would like, okay, thank you. Next, I would like to welcome our beloved principal sir and convener of today's seminar, respected Dr. Amol Joshi sir. Welcome, sir. Next, I would like to welcome today's program coordinator, Dr. Aruna Devi Viraja madam and Mr. Nadeem Khan sir. Welcome, sir. Welcome, ma'am. And lastly, I would like to welcome all the enthusiastic participants. Welcome, welcome one and all. Now, I would like to request Dr. Virajan madam to give a brief agenda of the today's topic. Please, ma'am. Over to you, ma'am.
Yes, good morning, all of you. I am here to introduce the brief agenda of today's seminar on the concept of PAT, that is nothing but the process analytical technology. It is and the important rule of implementation of the QBD or effective monitoring of the process parameters to produce the high quality finished product in the pharma industry. As it is a new concept introduced into the quality attribute of the different formulation and the pharmaceutical products, the application of PAT may be the part of control strategy included into the ICH guidelines, that is Q8R2 guidelines, which ensures the process remain within the design space. Now, PAT is the more effective for the detecting the failures. It also gives the multivariate data acquisition and the analysis. It is the process of analytical chemistry tool, continuous process optimization of the process monitoring control. It is preferably used in the pharma industry to get a quality product with the better design. So today we are organized the seminar on the same topic to get the detailed knowledge about it. For today's seminar, we have the two eminent personalities as the speakers. In the morning session, the Dr. Sachin Bhusari sir, who is working in the UDCT department, Dr. Babasaheb Marathwada University, Sambhajinagar. He is a very young and dynamic personality and a good scientist. Will share his knowledge of the PAT for us. As well as in the afternoon session, the Dr. Om Prakash Bhusnure sir, who belongs to the Channabaseshwar College of Pharmacy. He is working as a HOD and professor in the QA department. He is also the good speaker, will give the more detailed industrial approach of the PAT. As I am thankful to both of them for giving their valuable time for us, I am also thankful to our management, that is Chairman Sudhir Anna Patil sir, Dr. Amol Joshi sir, who is the principal of our college, administrative officer, that is Aditya Patil sir, secretary ma'am, that is Sau Prematai Patil, as well as all the peoples who are involved in this organization of this program. I'm also thankful to our institute to give me the opportunity to organize this seminar. I hope you all will enjoy the seminar and will get the enriched knowledge about the PAT. So thank you one and all, and welcome to all of you to join the this program. Now I will hand over the mic to Rajmane madam to give the detailed introduction of our today's first guest lecture. Thank you, sir. Thank you.
Thank you, madam. Thank you so much,
Over to
ma'am,
ma'am.
for thank you, madam, for giving a brief agenda of the today's seminar. Thank you once again. Now, with a great pride and honor, I would like to introduce a dynamic personality and the speaker of today's seminar, Dr. Sachin Bhusari, sir. He is currently working as assistant professor, UDCT, Dr. Bamu University, Chhatrapati Sambhajinagar. He is also a head of Center for Drug Metabolism and Pharmacokinetic Study, UDCT, Dr. Bamu University. Sir has area of research which includes preclinical drug development from ancient system of medicine and in analytical and bioanalytical method development and also in preclinical and clinical pharmacokinetic study and formulation development of biochemical assays. Sir have been granted with 3 Indian patents and one is filed. Sir has published more than 78 research papers in various international and national journals and published one book chapter. Also, Sir is worked on various grand research projects and consultancy, which includes 3 industrial projects, 2 government projects, and more than 23 academic projects. Sir has received various research grants with worth rupees in the crores and lakhs that includes 3.68 crores from Drug and Pharmaceutical Research Program of DST, Government of India in the year 2019-22, grant of grant of 20 lakhs rupees from CSR Government of India in the year 2019-22, grant of 19.30 lakhs rupees from DBT research project in the year 2014 to 17, grant of worth rupees 11.74 lakhs from UGC major research project in the year 2013 to 16, and grant of 24 lakhs rupees from DST in the year 2012 to 15. Sir has also received various professional recognition awards and fellowships, which includes Sir has been a recipient of National Facility for Bioanalysis. Sir is also a recipient of DST Fast Track Scheme for Young Scientists. Sir is also a recipient of Senior Research Fellowship of CSR Government of India. Sir is also gold medalist and stood first in the merit list of BPharm, and he is also best outgoing student and stood first in merit list of diploma. He is also working on various proportional positions like Sir is a member of Innovative Incubation Linkage, Dr. Babu University. Sir is a member of Research and Recognition Committee. Sir is a chairman of Ad hoc Board, Herbal Drug Technology, Dr. Babu University. Sir is a member of Ad hoc Board, Department of Chemical Technology, Dr. Babu University. He is also Ad hoc Board of Pharmacy, Dr. Babu University. Sir is a member of Department Research Committee, UDCT, Dr. Bamu University. Sir is also a member of Research Advisory Committee of UDCT and Government College of Pharmacy, Chhatrapati Sambhajinagar. Sir is also a uh, Sir is also a member of Research Advisory Committee of Ved Prakash Patil College of Pharmacy, Chhatrapati Sambhajinagar. So there is a lot more to tell about this, sir. So we are all very fortunate to have such an eminent personality with all of us. Once again, welcome, sir.
Thank you, madam. Thank you.
Thank you, sir.
Sir,
Now,
welcome, sir. Once again.
Thank you,
Yeah.
madam.
Yeah. Can start.
now before starting the seminar, I would like to request all the participants, if you have any question during the presentation, please type it in on the chat box. We will have a question answer session at the end. So now it's time to listen words of wisdom. I would like to request today's speaker, Dr. Sachin Bhusari sir, to start the session. Please, sir. Over to you, sir.
Thank you, madam. Thank you for nice introduction. Thank you.
Thank you. Thank you, sir.
So, a very good morning to all of you. Thank you for such a nice
Good
introduction.
morning.
Am I audible?
Yes, sir. Yes, sir.
Yes. So thank you. Thank you so much. So at the first, I would like to offer my sincere thanks to KT Patil College of Pharmacy, Dharasiv, for arranging such a nice session. I am thankful to Dr. Joshi, Dr. Birazda ma'am, all the entire team of KT Patil, as well as the owner of the KT Patil Pharmacy College. Thank you for giving me an opportunity to share my views on this PAT. So let us start with this process analytical technology PAT concept to applications. So my dear students, PAT it's very new, innovative, and very recent approach which has been launched by USFDA. So it becomes necessary to keep ourselves updated in this domain because we all are practitioners of the pharmaceutical technology rather. So it becomes necessary to have a deep understanding about the forthcoming concepts that are widely used in industry. We all are aspirants, so it becomes necessary. So this is very new area, new techniques, new framework, and by through this session, I will try to explain my views, my understanding about the PAT. So it's concepts to application. So my presentation will cover almost basics to its application. So now, if you look towards the scope of the presentation, today's presentation, so you can see nicely. Uh, for the PAT, I'll be covering historical perspectives, timeline of the PAT. So from where this concept started, basically it becomes necessary. It becomes necessary to understand the origin of this PAT. Then I'll be covering some official guidelines and agencies involved in this PAT, the backbone of this PAT. Again, goals, characteristics, and challenges of the PAT. My presentation will give you an overview about the product versus process approach. That is the latest one, which one has to understand. Then I'll be covering PAT framework in detail and the tools which are used in PAT. Then I'll be covering PAT in API as well as formulations because in pharma industry, basically it is not a synthesis of API only, and it is not limited to tablets and capsules. It covers everything: synthesis of API, formulations. Nowadays, we are more into impurity profiling, impurity manufacturing, degradation product manufacturing. So, PAT is applicable in every segment: API, formulations, impurities, and everywhere. Okay, so I'll be covering. I will try my best to cover these sections also. I have some data about the companies which offers these services because PAT is kind of service. Basically, it's a complete package which has to be designed properly as per the individual need. So I'll be trying. I will be covering some companies which can offer these PAT services. It is needed because some of you might have that background. Some may have chemical industries, pharmaceutical industries, or someone may have that ability to develop that companies. So one should know the PAT services, who offers the PAT services. Now the next part which I will be covering in my presentation will be universal analytical tools in PAT. Because in PAT there are various critical tools which has to be managed nicely to give output or to get the output. So my presentation will focus on NIR, Raman, UV, HPLC, MAS, FBRM, NMR, and ARS. As a pharmacy student, I am pretty sure that UV, HPLC, MAS, NMR, these are the four basic tools you might have well aware of. But as far as FBRM, ARS, Raman, near IR is concerned, yes, we are little bit into that. So my presentation will cover that section also. Now, some of the PAT applications I'll be covering in that, and beyond that, I'll try to give you some glimpses about the validation. So this validation parameter has specifically added in my presentation. Basically, we discussed with organizers, and it was a requirement. Yes, sir, if possible, try to give some glimpses. So instead of method validation, I'll be more towards instrument validation because method validation you all might have a backbone, but for instrument validation, yes, you pretty sure you need some core information with which you can optimize or with which you can calibrate the instruments. So I'll be covering that part also. So this will be our scope of today's presentation. So let us start with the introduction. So before going into PAT, basically one should know why the PAT was there, what was the reason why it was there, and how it came into picture. So the origin of the PAT lies into health system, human health system. So we all are pharmacy background people, and we know in this world pharmaceuticals and food, these are the two sectors which are very close to our heart, body, mind, because these two sectors directly impacts the health of the human beings. Am I right? So in that case, it becomes we all are more concerned about the quality of this food as well as pharma. So I'll be focusing my discussion for the pharma itself, but you can understand, you can pretty well consider that food is also same part of our discussion. But for today's session, I'll be more towards the pharmaceuticals. So when it comes to pharmaceuticals, especially formulations, you can see as picture shows injection vials, ampoules, capsules, tablet, and all those stuff. So when we try to purchase any medicine, we are more concerned about the quality of the medicines. Reason being, we know we always take the medicine whenever we are in that ill situation and we want to get rid of that immediately. So quality is a very concerned, genuine matter in the formulations. So we look towards the quality of the medicines and we try to purchase quality medicines. So previously, even right now also, we are more towards the branded formulations or the pharmaceutical preparations, because we do have that concept that anything which is branded, it's a quality medicine. So last 10 years now scene is entirely different. We do have good generic medicines also, but yes, definitely, let it be generic, let it be branded, we are more concerned about the quality of the medicines. So if you see the concept of quality in pharmaceuticals or formulations, APIs, so conventional approach, we were more towards quality testing. So we do have good background pharma, all of us. So what we learn basically from the theory, what we practice is quality testing or quality control. We try to control the quality of the formulations or API. So till 2000, it was basic concept to test the product for its quality intermediately as well as at the end. But last 23, 24 years, the scene is entirely different. Now, if you see unconventional part of this quality consciousness, now we are more towards quality building. We are trying to build the quality in the preparation, not testing the quality, not controlling the quality. And that was the origin of PAT. So why PAT came into picture? We changed our approach from conventional to unconventional. You can say conventional to unconventional. Since 2000 onwards, quality building came into picture, and that was the initiating or initialized point of PAT. So this is background of the PAT. So let us have a detailed look into PAT. So what is PAT? As all of us we know, it's a process analytical technology. So that means there are three words: some processes involved, some analytical techniques are involved, and it is a technology, basically management of all this process as well as analyticals. So if you look into this graphical representation or picture, so you can see at the left side, it's a process which is coupled with some analysis. And if you try to manage all this process with some analytics, so then it is something like PAT, process analytical technology. So try to understand just that PAT is we do have some technology through which we are trying to manage some processes with the help of some analytical tools. It's clear understanding. I'm sure some of the PG students will be there, so I'll be trying to explain some of the basic things also, not only applications but some basic things which can be useful for you people in your PG programs also. Some of the PhD students might be there, so they are also more interested. They might be interested in this information. So what is PAT? It's process analytical technology. It's a combination of process and analytics and the technology to manage all both process as well as these analytical tools. If we do that, that is PAT. So this is for understanding. Even US FDA has clearly mentioned and defined the PAT. So you can see a system for designing, analyzing, and controlling manufacturing through. There are two highlights in this definition. Timely measurements. The best part of this PAT is real-time measurements. Of what? Of critical quality and performance attributes of raw and in-process materials and the processes with the goal of ensuring final product quality. So in this pack, do you know it's good thing to understand. It's a real-time measurement just for ensuring the final quality of the product, final product quality. So PAT is the best tool for this timely measurement and ensuring final product quality. So this is the classical definition which has been given by the US FDA. Now, yes, we do understand what is PAT, classically how to define PAT, what are the key, what you can say, full forms of the PAT. But it becomes necessary to understand some of the historical perspectives of the PAT also, how it came into picture, what was the reason, what was the need, and how it gets developed. So if you look into that conventional approach, so previously we were more towards manufacturing, processing, and intermediately testing the samples. So you can see here conventional pharmaceutical manufacturing is generally accomplished using batch processing. So we do manufacture the formulations in the batch, 1 lakh tablet, 2 lakh tablet, yes. And we do have backbone of laboratory testing, quality control testing is QC lab is there, QA lab is also there in the pharmaceutical industries. So that is a conventional approach, which is something like offline, wherein you are manufacturing something and you are testing that outside your manufacturing area or else adjacent area, but which is not a part of manufacturing area. Okay, so that was a conventional approach. So further, it was thought was given that is there any scope of improvement in this conventional way? Yes. So people found that yes, there is a significant scope of improvement, significant opportunities. Basically, there are significant opportunities to improvise. What to improvise? Pharmaceutical development, manufacturing, quality assurance through innovation in product development, process analysis, and process control. All the four arms of this manufacturing quality assurance can be modified, can be improvised. So that was the thought, and there was a scope. So based on that, again there was one technical hitch of the regulatory uncertainty. As you know, pharmaceuticals as well as food sector, it is regulated sector. We all are bound to regulatory authorities, and now it's very simple. Every authority, just like BCGI or USFDA, we don't have that kind of projections. What guidelines will come into picture and what has to be implemented immediately. So previously it was thought that we have to follow only the guidelines which are given. We have to stick to that guideline, and there is no alternative. We cannot change it, and that was the constraint from the regulatory point of view. But afterwards, in nearly 2000, that thought was changed by the agencies itself. And the leading agency which molded this concept of rigidity was USFDA. So you can see potential recognition. USFDA identified that yes, although we should be strict somewhere to manufacturing quality control, quality assurance, but they thought that there should be some innovation, and they initiated that innovative process through this CGMP guidelines, current good manufacturing guidelines. So pharmaceutical CGMP for the 21st century risk-based approach was launched by the USFDA, and that was the starting point wherein the governing agencies, regulatory agencies thought that yes, we should change our perspective. So PAT, if you go through the history of the PAT, you will see this flow wherein the PAT was changed. So now we are looking towards the PAT. So it is basically need to understand the timeline of the PAT also, how it was developed. Although my slide represents here as the starting point of 2001 to 2004, that was the launching period of the PAT. from its official introduction to guidelines. But I would like to share some information with you people. PAT is not new for us. The term was coined in 1990, basically. That was the year in which the term was coined, and that was official in 2001. PAT as a term, but conceptually, we are far behind that. Rather, if you track the history of the PAT in generic way, so you will find that some of the industries were using this PAT not as a name of the PAT, but some principles of the PAT. Even 1900 centuries, 100 year back, if you track the history of brewery industries, Gossett and all, so you will find that people were using the mathematicians to improvise the processes, and that is a key attribute of the PAT. Now, in my few next coming slides, you will come across that. So, what are the critical attributes of the PAT? So, here, there you will find this mathematical backbones. So, 100 years back, even industry started to utilize some of the extraordinary personalities, expertise, which are not from the production, which are not from that manufacturing processes, but they are giving some inputs for the process optimization, process modification. So PAT is not new for us, but as a term, it was introduced in 2001. That is the some background. That is for your information. So USFDA, there is one advisory body of the USFDA. USFDA, you know, FDA, Food and Drug Administration. So it covers both the arms, food as well as drug. So for the drug, there is one technical advisory committee for the pharmaceutical sciences. So that advisory committee initiated this PAT in 2001. So they came up with some this PAT concept, and they launched this term. And in 2002, they come up with broader perspective to enhance and modernize the regulations of the pharmaceutical product quality. You all of you know that USFDA is the largest single hand agency which almost governs all the pharmaceuticals all over the globe. Basically, they are into very well manufacturing, good quality product manufacturing, and they are the designers of the guidelines. If you follow that, USFDA, I think my knowledge says that if you stick to USFDA guidelines, you can crack any audit. That is the scenario in India also, because they have that stringent guidelines. So they come up with some modifications in regulations of the pharmaceutical product quality in 2002, and initially in 2003, PAT draft was published. Publication of the PAT draft was in such a manner that if you have some suggestions, you can give some inputs. It was for public domain, and from public domain, it was expected that there would be some inputs on these guidelines. Yes, definitely there were inputs from all over the globe, and finally in 2004, PAT guidelines was introduced. So see, this is the guideline which came into picture in 2004. So if you critically see this title of this guideline, it's very nice. Basically, it's a framework. They are saying it is a framework for innovative pharmaceutical development. So for any innovation, previously it was not that was not a for we. It was not a free home ground or playground to do the R&Ds. But now they are into innovative pharmaceutical development. So guideline itself says that you can have that innovation in your pharmaceutical development. You can have that innovation in your manufacturing also, and quality assurance also. So that was the beautiful part which was introduced by the USFDA in 2004. So now USFDA was ready to have this liberty, to offer this liberty to the industry. So if you follow this guideline, if you follow this framework, so you do have opportunity to innovate in your development, manufacturing, as quality assurance. I would like to make some comment in this slide. Basically, while discussing this, all of us very well know that if we have any product with us, if I am having my industry, I have to register my product. Product registration comes along with all the process registration, equipment registration, and everything. So whatever is I'm manufacturing, it is registered somewhere. So I cannot change by myself. Correct. So now through these guidelines, it was clearly indicated that if you use the PAT framework, so you can have a chance to modify your processes, manufacturing processes, even your API synthesis processes. You can change your quality perspectives also. You can change your quality parameters also. Okay, so that was the initiation which was done by the USFDA in 2004. So now again question comes in mind that only the USFDA was the agency who was offering this PAT, or rather, who is behind this PAT and who is giving us the guidelines of the PAT. So, as far as regulatory agencies are concerned, yes, definitely USFDA is the largest agency which has given us the guidelines for the PAT. But along with this USFDA, there are three, four regulatory agencies which are in favor of this pact. One is ICH. I purposely wrote here the full form of International ICH, International Council on Harmonization of Technical Requirements of Pharmaceuticals for Human Use. Those who are in the studentship, UG, PG, so some may need this information. ASTM, American Society for Testing and Materials. I would like to talk about ASTM also. EMA, European Medicine Agency. Those who are in PhD, PGs, those who are in this active project dissertation part 1 and 2, see, we do use some standards. We call it as analytical standards, but there are some standards which are traceable. ASTM standards, NIST standards. So these are the standards which are traceable. If you work with these ASTM or NIST standards, so your process or your development can be traceable development. So try to use this ASTM or NIST traceable standards in your dissertation. So that is the additional information I would like to share with you people. Regulatory agencies are concerned. So these are the four agencies which are rather more in the favor of PAT. Apart from this, there are some public domain organizations which actually supported PAT like anything. So you can see CPAC is there, CPAC is there, ISA is there, ISP is there, NIPET is there. So National Institute for Pharmaceutical Technology and Education. So ISP, ISA, CPAC, all these agencies, if you look, except EMEA, all these agencies are US agencies which are advocating the PAT, because definitely PAT is the upcoming tool, and basically not tool, basically framework, but it will help to build the quality in the product. So regulatory agencies along with some public private domains, the PAT is supported like anything. So up till now we were into what is PAT, who launched the PAT, what is the historical perspective, how it got introduced into the domain of the regulatory part of pharmaceuticals. So now it becomes necessary to understand what are the goals of the PAT. Why PAT? Why PAT? Why to go for it? So if you read this slide, you can understand. Basically, it reduces the production cycle times. So the goal of PAT was to reduce the production cycle time. Now, if you ask any industry people, do you know the wealth of the industry people? It is not money. It is first time, then money. Because you can earn the money at any later point of time, but you cannot earn the time. You cannot manage the time. Basically, you can get the time. Whatever time frame is available, you have to work in that only. So in industry, if cycle time is less, no, it is as good as full profit. So reduction in the cycle time is the one goal of the PAT. Okay. Second goal of the PAT is preventing rejects, scraps, and reprocessing. My dear friends, just consider one case. You manufacture 1 lakh tablets, and now after analysis of tablet assay, you find that assay is less, deviating from my standards by 1%. What to do? It's a reject. Basically, it's a reprocessing. It goes into scrap. So it's directly waste. So if you are into real-time monitoring of this formulations API, so that reject, scrap, and reprocessing can be avoided. So second goal of the PAT is preventing reject, scraps, and reprocessing. So you can basically saving the money, saving the time, basically saving the time. Third goal is real-time release. Now in batch processes, what happens basically, we synthesize API, or in batch, or we manufacture tablets, capsules, and we submit the samples for the QA/QC, and then we analyze the material, and then we release the batch. It is a offline basically process. If you have a PAT with you, so real-time release is quite possible, because whenever there is a manufacturing of this particular batch, say suppose tablet, paracetamol tablet, if you have real-time monitoring, real-time analysis through PAT, so you can release the batch immediately. No need to take 24 hours or 48 hours. So that was the another goal, because industry is on the toes always. Now fourth is increasing automation. Nowadays, if you look into industry scenario, to get the well-trained manpower itself is the biggest task, biggest issue. So automation is highly preferred in all the industries. You consider automobile industries, you consider pharmaceutical, food, every industry, whatever is the scenario, they all are more towards automation, because human error is there definitely. And if you automate the process, you can reduce the human error. And if you reduce the error, that means you can get the good quality material. Okay, so automation is quite possible by using PAT. So another goal of the PAT is automation. Now, fifth goal is improving energy, material use, and the capacity. That is another goal. Basically, if you reduce the scrap, rejects, reprocessing, definitely you are utilizing the energy as well as material very effectively. 100% input can be converted into 100% output. If you talk about in terms of percentage, no. So it's quite possible, even for energy, light, man hours, water. These are all energy resources, no. So if you try to manage that, if you try to reduce the wastage of that, so you can earn the money. Even capacity. If my plant is having capacity of 10 lakh tablets in 8 hours manufacturing time, and if there is a zero rejection, yes, I am utilizing full capacity of my plant. So PAT goal is improving energy, material use, and capacity also. And next to that is continuous processing. Now we are more towards continuous processing. That there is one concept I will talk to you regarding that also. CM, continuous manufacturing. Continuous manufacturing. Although it has a slight relevant on this slide, not next slide, but this slide. Continuous processing is another goal of the PAT. If you have PAT framework, if you have PAT technology, you can into continuous processing always. So that means what? You can have continuous manufacturing, you can have continuous testing, and you can have real-time release. So you can keep your plant 24/7 on, and you can earn, and you can go for manufacturing as well as whatever your task. Say maybe API, maybe formulations, maybe impurities. So these are the goals of the PAT. Now, what are the characteristics of the PAT? So if we know the targets of the PAT, now what are the attributes, what are the characteristics of the PAT? It is necessary to understand. While before going into PAT, one must have. So multivariate tools and measurement systems. This is a typical peculiar characteristic of the PAT. So PAT framework has such tools, measurement systems. So where we can have analysis of more than one variables at a time. You know, process or product, there will be multiple variables, more than one variable. So if you try to test one variable at a time, so it will be difficult for you to optimize. So multivariate tools and measurement systems are the characteristics of the PAT. Now, real-time data linked with process control system, that is another characteristic of the PAT. Basically, PAT framework, if it is applied by understanding the processes, so real-time data you can achieve, and you can have that real-time data linked with process control. A classical example is, if you are synthesizing any API, just consider, and you need pH of 7.4. Okay, and now your probe is there in reactor, and it is showing pH 7.1. So you know it. Basically, it has not been achieved. 7.4 pH is not achieved. Real-time data. So what to do? Adjust the pH in such a way that now it will be 7.4. My dear students, in industry, it is not simple task. It is quite possible in laboratory to adjust the pH with our small pH meter and 50 ml volume. When it comes to 1000 liter, 5000 liter, 10,000 liter glass line reactors, it is quite challenging. So what we need is real-time data. So PAT has that facility, real-time data, and that has to be controlled with process control system. So unless and until there is a pH 7.4, that process will not get initialized because PAT framework is such that you have that control system. So that is another characteristic. Now, third is application in numerous pharmaceutical sectors. PAT is used in the various sectors, right from manufacturing of API to packaging also. So now you can visualize the aura of this PAT. Now, PAT is basically another characteristic. It is backbone or supported by QBD and CM. Yes, I was talking about continuous manufacturing previous slide. So it has relevance here. So PAT is having a backbone of QBD, quality by design. Now we are more towards designing the product in such a way that we can get the quality. So now the concept is designing the quality, not controlling the quality, and that QBD approach is the peculiar characteristic of the PAT. Now, improvement of the process. So PAT has another characteristic. If you want to improvise real-time process at actual during the manufacturing, it is quite possible with the PAT only because it is very simple. If you have PAT framework, then only you can get the real-time data. So it is possible to improvise the process only when you get the real-time data. Correct? Am I right? And another characteristic of PAT is real-time product release. So it is quite possible if you have raw data with you, which is generated by your PAT. Okay, a process is ongoing. So after just finishing the batch, now you are very sure that my batch is having this much assay, this much crystallinity, and everything. So you can release the product at in real time. That is another characteristic. And integration of the systems. PAT is having the integration of all. So my presentation will cover that also. So I'm going to talk about integration of the systems also. So there is one view, design expert. It is a software, experimental design software. Design expert is very well-known brand in design of experiments. So this is with me, license copy. So this is a screenshot of design expert. So those who are into masters or PhD, those who are into synthesis, those who are into development of formulations, please try to use this QBD approach, quality by design approach. This is a sincere request from my side. I am a supervisor also. So lot many times I come across situation wherein people are showing us the data with simply one factor at a time, change in one factor at a time. So being a pharmacy student, being a student of pharmaceutical technology or industry, so you should have this mathematical backbone. Now, although we know the characteristic, we know the goals, now it becomes essential to understand the challenges also because PAT, it is a new technology, it is a good technology, it is a new framework which is giving us good back as an industry. But there are some challenges. Everything has both the sides, pros and cons, advantages, disadvantages. So in this case, yes, we do have some challenges in PAT. So before applying, just consider that you have industry and you are trying to apply PAT for this discussion, for this slide. So now, if you are into PAT framework, you are trying to implicate the PAT. So we don't do that blindly. What we do is we go for homework. Am I right? So in this homework, what to do? Just try to understand challenges. PAT is entirely segregated system. It is not a conventional system. Every industrial need will be different. So that's why PAT service offering agencies are different. As per your own need, you have to design the PAT. Process analytical technology, if you want to implicate in your industry, so it is a segregated system. It is not an integrated system. It is entirely different system from our routine quality control, quality assurance. So that is one challenge. Second challenge is till that it is not mandatory. Implication is not mandatory. US FDA has given us guideline. Yes, definitely. A guideline means if you want to be in the race, you have to keep yourself updated. Correct. So PAT guidelines are there. Every industry is trying to follow the PAT, but it is not mandatory. That is another challenge because if it is mandatory, we try to implicate it, we try to use it, and if there is a mandatory implementation, then there are developments. Then only there are developments. So the another challenge is it is not mandatory. Third part is tool data and data analysis different from traditional methods. In PAT, basically, my presentation will give you the idea about offline and online tools. That is the classical part. You must watch, you must see. That is there in my presentation. Yes, I am going to share it with you people very soon. So tools which we use in offline and online are different. Data analysis also it is different from traditional methods. In our laboratory, what we do is we get the UV spectra, we get the IR spectra, we try to read it. That is different story. But when that data is generated online, inline, then the data is slightly different and interpretation of that data is slightly different. Because online data, it is not a manual data analysis, it is automated data analysis. Try to understand. Okay. Now, third challenge is there is no historical precedence in industry. See, if PAT is, if just consider it is overall worldwide, it is available and everyone is following, then it becomes easy to implicate that PAT in our industry because we know how to do it, what are the tools and what are the constraints. Right now, there are very few industries who are into PAT framework, who are applying this PAT in their processes. So we don't have that backbone support. Reference material, we call it as a literature, no? Literature. So we do have limited literature. That's the another part. Then another challenge is data processing models needs continuous update because whatever data is generated from this, every process is different, every manufacturing is different. The kind of data which is generated, it needs updates. That is another challenge because you have to be on the toes for these updates. Now method validation platforms are unguided. Conventional, if you see the method validation parameters, so analytical, bioanalytical methods, we do have some defined set of guidelines. We do, we basically follow that guidelines and we just replicate the guidelines. We try to follow that guidelines and we get the data. Method validation, 3-day validation, intraday validation. Okay, for bioanalytical, tabletop validation, freestyle validation. So these guidelines are directly available, but these method validation parameters are offline. When you are using any method online, the validation of that method, it has some deviation. So that guided platforms are right now not available basically. That is another challenge. Now, one of the biggest challenge in this is unless and until verified, you don't rely upon that data because acceptable performance is quite questionable. It becomes difficult to verify. So for PAT framework, whenever you are applying this PAT framework, no acceptable performance, whether you are getting it or not, it has to be defined one, and it has to have some specifications, limitations, and that are not available right now. Now another challenge is basically in PAT we always focus on A in between word analytical. So we are more towards analytical oriented, more towards instrument oriented. Analytical is only one part. Basically, it has a process as well as technology. And analytical is only one tool. My presentation will cover that also. Now, that is misinterpretation. Basically, we should consider both PAT, all the terminologies. Now, data collection, implementation, and storage as per regulatory compliance. See, whenever we collect any data, I'm sure some of the industry friends will be there in this presentation. We collect the data, we store the data in hard copy, report formats, even soft copy, and it is stored. Okay, but this is offline data which is generated after some time. When the data is online, how to collect it, how to interpret it, and how to store it in compliance manner. 21 CFR compliance system should be there, and it has to be linked with the regulatory requirement. So there should be some regulatory guideline which will tell us, sir, collect the data in this format, interpret it in this format, and store it in this format, then only it is valid data. That kind of regulatory compliance, it is right now in PAT, it is limited. It is limited. To the best of my knowledge, I'm saying that it is limited. Now, approval to changes in existing products. We all are in approved products. You know, if you want to change any little bit, even polymer, if you want to change excipient, it is not in your hand. Once you submit the product, if you want to even change the excipient also, you have to take a permission. You have to fully validate every product every time. So changes into post approved changes in existing product, it is difficult, and PAT tells you that only online data. So at regional as well as global level, if I am having a US FDA compliant plant, and if I am trying to have PAT framework right now in my industry, now there will be difficulty in getting approvals from the products for the product by US FDA, even by DCJ also. Don't just talk about DCJ, highest authority in India. If you, I want to change my product slightly also, I have to seek the permission from the DCI in written, and it's challenging and difficult task. So that is a real challenge in PAT also. And another challenge in this PAT is, even if you consider a same agency, you consider any pharmaceutical organization, in that operational and organizational structures are different. Even for a same company, operational and organizational structures are different. So reason is that why it is a challenging PAT, the mindset. My organizational structure of this Aurangabad Chhatrapati Sambhajinagar plant is different, and same company if I am having in Gujarat, so I might have different task. So Chhatrapati Sambhajinagar and Gujarat, Bododara, it won't matter much. It's only zone difference. The PAT framework should be same, but while implementing that, it will be difficult. Okay, so there are some challenges. So now let us go for this product versus process oriented approach. This is typically for PG students, MPharm, MTech students, and even UG students. So previously we were into conventional product oriented analysis. If you see the slide nicely, the A part, starting material is there. You do some processing, you get intermediate. You do some processing, process two, you get bulk material. You do some processing, you get finished product. So we were more towards testing the quality of intermediate, bulk material, and finished product. That was the conventional approach. It was product oriented approach. We were more interested into product. So we were trying to test the quality of that intermediate, bulk material, and finished product. But now to 2000 onwards, now we are into process driven approach. What is that? Now we consider intermediate, bulk material, and finished product. Yes, definitely. But before that, we are more focused towards process one, process two, process three, and we are now trying to monitor the processes one, two, three. And PAT helps you there in process monitoring. And if your processes are correct, your critical parameters are within the specified limit, definitely your whatever you are getting, intermediate, bulk material, finished product, definitely it will be within specified limit. Correct. So that's the reason now we have shifted from this product oriented approach to process oriented approach. It is for understanding. Although we are testing the products right now also, but now we are more towards process driven. So now let us see the PAT framework. So backbone of this PAT is quality by design. Quality by design is the backbone for this. But before going into PAT framework, one should understand there are two critical steps. First, one should understand the process nicely. Whatever process is there, process means not only synthesis. Don't get confused. Basically, process can be of anything. It can be a synthesis of API, it can be a manufacturing, it can be anything. This is a process. So first try to understand the process, and step two, now then apply some principles and tools. So while understanding the PAT framework, first step is to understand your own process thoroughly, and then to apply the PAT framework principles and tool into that process. So slide classically explains this. So there are four critical PAT tools. You can see. I think I can show that in by using my cursor. I don't know exactly. First time I'm using this tool. So multivariate tools are there. So design of experiments are there. Multivariate data analysis is there. Multivariate processing parameters you can see. So multivariate tools is one part of the PAT tool. Analytical instrumentations, online, inline, outline, offline instrumentations are there. Yes, I am going to talk about that also. That is another tool. Third tool is process control tools. So we can have this process control tools. And fourth is continuous improvement and knowledge management tools. So there are four critical PAT tools. If you have this understanding of this multivariate analytical instrument, process control, and continuous improvement and knowledge management tools, so then to understand PAT framework is quite easy. Even to apply the PAT framework becomes easy. So first of all, one should understand the process and then principles and the tools. I would like to tell you about this QBD in this slide, quality by design. So now PAT framework has a backbone of quality by design. QBD is a very frequently used term by all the industrialists, even researchers, because it has gained a lot importance in today's scenario. So now we are into designing the quality, quality by design. We are designing the experiments in such a way that whatever we are achieving, it's a quality. And PAT framework is based on that only. So QBD hand to hand PAT framework will give us a good quality product. So product and process understanding and process control, it is a center of this PAT framework, and it is supported with all these four different tools of the PAT framework. So one by one, now we will see these tools. So first of all, before going into the tools, I would like to make some comments on this process understanding. So well understood process. So there are three arms of this process understanding. So how rather I would like to express that how to understood the process. Let us go in this manner. First of all, one should identify the critical sources of variability. In my process, what are the variables? Not only variables, what are the critical variables? See, if you are preparing tablet, if you are into tablet manufacturing, yes, definitely my binder percentage, my compaction force of the machine, yes, definitely glidance, okay, ready to use granules, the quality of ready to use granules. So all these are critical sources of variability. So I must have understanding of my own process. If you consider the API, pH, temperature, agitation, RPM, even it matters a lot. If you work in the laboratory, if you are into pro-liposomal manufacturing preparation, if you are into microsomes, pro-liposomes, if you are into especially what you can say is pellet formulation. In pallets also, single screw extruder, multi screw extruder, while jacketing equipments, single screw extruder. If you have jacket, water jacket, cold solution you can run, you can make it hot process or cold process. So all these are the critical process parameters or product parameters. So one has to understand that first. Now, second arm of this understanding the process is one has to have a management of the variables by the process. I should have some process with me so that I can manage the variables. I know there will be some variation in my process. So how to manage that variation? So that mechanism I must have. So it comes under understanding of the process also. That is the second part. And third arm of this understanding the process is I must have, I must know the final product attributes, final product quality attributes, whether I am getting the 99.9% assay, whether I am getting the free flowing material, whether I am getting the material with crystal size of this much size, very specific, very specific, whether I am getting the CARS index, flammability, and all these parameters. So my final quality attributes must be known to me. So it starts with variables, variable management, and the final product quality, final product quality. And if I understand all these three arms, no, then I one can say that I understood the process. That's understanding the process. So got my point. Let us go to next. So now, after understanding the processes, now just try to understand the PAT tools. PAT tools. So there are five critical PAT tools. Five critical PAT tools. So one PAT tool, I purposely segregated these tools into continuous improvement and knowledge management tools, four and fifth. So if you track the data, if you dig out the data, you will find that in some databases, continuous improvement and knowledge management tools are merged together. So there are reasons for that. Continuous improvement, the input from continuous improvement goes into knowledge management tools, knowledge management system, and both these segments. But for understanding, I purposely segregated this into continuous improvement and knowledge management tools. So in multivariate tools for design, data acquisition, and analysis, that is one tool. As I say, to understand the variables, to manage the variables, okay, to find out the criticality of that variables, to optimize that variables, one must have a backbone like experimental softwares, experimental design softwares are there, data analysis softwares are there, data acquisition softwares are there. So this tool, specifically multivariate tool, it is more towards software based, more towards software based. Now, second tool, PAT tool is process analyzers. This is very critical as far as PAT is concerned. That A stands for AA in PAT, that is analytical, and that's process analyzers belongs to this A, central A, process analyzers. Third is process control tools. So if we analyze the data, we know the variation. Now how to control this? So there should be some mechanism by which we should control the process. So control tools are there. And as I said, continuous improvement. So PAT, as it is an online tool, it gives you the continuous data, real-time data. So there is continuous improvement in the process. And that data, that information goes into knowledge management tool. So that's why there is another segment that is called knowledge management tools. So now just see multivariate tools for design, data acquisition and analysis. There are some changes in my presentation when I'm presenting in this specifically StreamYard. There are changes in this what you can say is graphs as well as the figures as well as some textual matters. That is slight change because of the unit which we are using. Okay, so that is just forgive me about that. It's a change because of this module. Okay, so now multivariate tools for design, data acquisition and analysis. So there are 4 steps basically in this. First of all, you have to have a experimental designing for your process. You may have surface response methodologies, process simulations, pattern recognition tools. So first step or first part of your multivariate tool, PAT tool, is a backbone, statistical backbone for your process. Second is you must know the orthogonality, reference distribution, randomization. So that means variation. Variabilities must be identified statistically, and one must have understanding about these variabilities. Now, third part is identification and evaluation of critical performance parameters. In multivariate tools, the third part, one must have a identification and evaluation of critical performance parameters. That means the factors which are critically affecting my final product or process. Okay, that has to be identified. And the fourth part is after this, if there is some failure, if there is some defect, so why this failure or defect is there and how quantitatively it is impacting my formulation or the product quality, that has to be identified. Why? It's very simple. Unless and until you know the source of failure, source of disqualification, how to correct it. So multivariate tools will give you this backbone. So we do have experimental design softwares, we do have randomization, we do have critical performance parameters, and now the inputs from the failures also. So this is one tool of the PAT, which is distributed into these 4 segments. Now, I would like to make some comments regarding this process analyzers. A from the PAT. A is a letter from the PAT, PAT. The A is there, analyzer. So this is typically the analytical tools which we use in this PAT framework. So broadly, if you want to classify the types of measurements, so it is offline. My dear students, we generally practice this offline testing in your laboratory. You do some dissolution test in your laboratories by using dissolution test apparatus. You collect the sample, filter it nicely, 4 hours, 8 hours. After that, you switch on the HPLC, set the HPLC, then you analyze it. If you don't have HPLC, you go for UV and then you analyze the samples. Dear students, for DISO and any purpose, UV is not a suitable tool. Okay, so you must go for HPLC. Side by side, I am trying to give you some glimpses wherein what it can be done and what cannot be done. Okay, so just try to remember this. So this is offline tool. That means what? It is not coupled with process of manufacturing, process of testing or anything. It's an offline tool which we use it, which is near to your laboratory or another segment. Now, online tool is something like, if this is the product flow, so your analytical tool is based in such a way that product flow is diverted, it is going towards your instrument, and now again it is coming into circulation. That is called online mode. So there is one loop which is created purposely, and in this loop there is your analytical tool, that is called online tool. We casually use the term online. This is the real meaning of online for the PAT framework. PAT framework. Now, for inline testing or types of measurement, your instrument is in line with the process. It is the part of the process. It is a loop. For online, it is a loop. For inline, it is inside the process, inline. So, if the product flow is from this direction, it will pass through the instrument, it will go. It will be a continuous flow through instrument, and it will be called as inline. And third part which is there, that is called at line. So at line is what? Your product is there, product flow is there, and near to that, the testing equipment is there. So it is at line. So most of the industries now, right now, which they follow is at line. For academic, yes, definitely offline, but for industries, right now they are into at line, and some of the industries are now on this platform, online platform. So now clear. So types of measurement: online, inline, at line, and offline. Now see the process control tools. So in process control tools, so third tool of the PAT is process monitoring. Monitor the state of the process and manipulate it to maintain the desired state. So we do have some process monitoring tools, software based tools, which will indicate you, yes, pH is not correct, modify the pH. Yes, temperature is not achieved, just raise the temperature by using heaters. So these kind of process monitoring tools are there, and that tools will help you to get the optimized values. Okay, clearly understand? Now the control strategies. So again, there are some process control tools which helps you to control the processes, attributes of input materials, reliability of process analyzers, and achievement of process endpoints. So these control strategies, say once you achieve the product, it will stop the reactors at a time. This is a control mechanism because it is a real-time monitoring. So product is achieved, you got the desired product, yes, definitely there will be stoppage of the process automatically. So control strategies are there. Now in that, it will not only it will not only stop the processes, but it will help you to identify the quality of the starting material, the parameters which you set, and it will control that. So that control strategies are integral parts of the process control tools. Now, this is one of the classical way of steps involved in design and optimization of drug formulation within the PAT framework. So now in PAT framework, if you see identification and measurement of critical materials process attributes relating to product quality, it is coupled with design a process measurement system, real time and near real time monitoring. Again, it is coupled with design process controls that provide adjustment to ensure controls. And lastly, to develop the mathematical relationships between the product quality attributes and measurement of critical parameters. So now continuous improvement tools are there. So continuous learning is needed. PAT gives you that chance to get the real-time information, continuous learning. It justifies post-approval changes and identifies vital products and process. Now in continuous improvement, there are 3 to 4 different tools. I am going to talk about that. ERP, Enterprise Resource Planning tool is there. EMS tool, MES tool is there. Manufacturing Execution System is there. Plant Maintenance Report, PMR is there. Deviation reports are there. And some of the knowledge management tools are there. Kaizen, Lean, Six Sigma. Just we'll have this. So see, ERP tool is there. It is one of the classical tools wherein you can have this human resource management, planning, inventory, reporting, CRM. Then you have manufacturing execution system is there. First of all, you have to manage the resource, then material management, personal management, data collection, quality management, information planning, production planning is there. So these tools are available for this Now we do have Six Sigma, Kaizen. Kaizen is Japanese technology, Japanese framework which is given by Japanese people. It's for just to visualize the problem, develop countermeasures, root cause, find out root cause, hypothesize solution, test the solution, hypothesis, implement the solution, standardize work. Even Six Sigma. Motorola launched this concept Six Sigma: control, define, measure, analyze, improve. So these are the tools which are available in PAT also. Now let us see the PAT in API production. So this is Ian Clegg. This is a request to you people that just try to go through this process analytical technology chapter given by Ian Clegg. It's a classical way of explaining PAT. So if you talk about the PAT in API production, this is the classical framework. If you talk about API process flow, you need analysis wherein starting material has to be analyzed. So mid IR, near IR, Raman handheld chromatography tools are applicable. If your product is in reactor, the process is ongoing. So ongoing process can be monitored by calorimetry, mid IR, near IR, UV, Raman, NIR, polarimetry. Ongoing process. So analytical needs are there. Workup. When the reaction is completed, you have to work up the reaction. Synthetic people, they might understand very well workup of the reaction workup. So there what you need is NIR, FTIR. These techniques are used. Crystallization, specifically turbidity, FDRM, PVM, Raman, MIR. These tools are used for drying, milling. So all this from right from synthesis to finished product, right from the reactant to finished product. So there are a number of requirements at every stage, every process. So PAT will help you there. Now patent formulations. Similarly, previously the slide was for the synthesis. For even tableting, capsuling also you can see the PAT FTR instruments. So spray drying, hot melt exclusion, co-precipitation, co-exclusion, electrospraying. All these processes can be monitored by the probe, different probes. You can have FT Raman, if you can have FTIR instruments are there. You can see raw material mixtures, blending. Yes, my example will cover that blending also. So you can actually check the process and you can check the tabletability also. So PAT is in formulations also. So now this is one of the classical tool, PAT tool you can say. It gives you the active ingredient content of the tablet directly. So there are inbuilt technologies, NIR technology is built in that. So you can check the ingredients of the tablet, and you can have this online tool. Okay. So now let us see the companies offering the PAT related technologies. These are the companies which offers the PAT related technology support system: Kaiser, Malvern, Mettler, Thermo. So these are the companies which gives you the idea about the PAT technologies. So now this is the classical part which one must understand. Universal analytical tools in PAT. So what are the process analyzers? See, so near IR spectrometry. In near IR spectrometry, so it was launched in 1800 centuries. It's qualitative as well as quantitative tool, basically based on transmittance and reflectance. Okay, and the range of this is 782 2800 nanometer, 2800 nanometer. This is the working range. So offline tool, mid near IR tool. This is from Perkin Elmer. This is an offline tool, which will you can see the tool, and this is a spectra in which region it works. So near IR. I purposely kept this near IR in the first because in PAT, near IR has a very vital role, very vital role. If you dig out the history, if you dig out the literature, so you will find that near IR probes of near IR are frequently used in pharmaceutical industries nowadays. Handheld devices. How? Yes, I am going to show you. So this is the instrumentation. You can see light source, monochromators are there, samples. It acts on reflectance and transmittance. So the graph, actually output of near IR will look like this: wavelength versus transmittance. You can see this graph pattern. Now working of NIR. So this is the classical slide, very important slide. Now you can see the difference. Now it is a probe based. So you can use the near IR probe, handheld device, and you can take out this probe. You can take this probe to your API container, to your formulation. Any platform, you can take this probe with you. So NIR is connected to fiber optic probe. Probe is composed of optic fiber, lens, mirror, and signal channel. This is for student purpose. Transmission and reflectance on NIRs through the samples, and transmitted signal forms a spectrum through computer software. But you can collect the data by using this probe. Now the best part is how it looks like. Now can you see the difference? Tabletop instrument is looking different, but now see the difference. So NIR, when it is used as a PAT tool, see for the tablet machine, now this is a NIR spectrometer, which is placed in line to see the process. Okay. So at actual, the NIR tool, when it is coupled with equipment, will look like this. If you have this handheld probe, so you can take it to the API container, you can measure the content. Okay. Now Raman spectrometer. Raman spectroscopy is another tool which is frequently used. Raman spectrometer. It's a vibrational spectroscopy. All of you know it very well. So again, offline instrument will look like this for Raman. See, in general, vibration occurs in chemical bond. This is theory part. This theory part is purposely wrote here for the student, those who are new entrant in PG or UG. So they should read it. It basically characterized based on their molecular vibration frequencies. Okay. FT Raman, Fourier transform Raman. So how the instrumentation of the Raman will look like. So laser is there, laser beam is there, sample is there, lens detection system and computerized. It integrates the data. How it looks like Raman shift, centimeter inverse versus Raman intensity. This is the classical spectra which is published by Broker. Yes, mark is there. So for this aspirin and paracetamol, you can see overlapping spectra of aspirin and paracetamol. It is in single formulation, which has been generated by the Broker. So you can see that. Now working. So there is some display of working of this Raman spectrometer. For this is for student purpose. You can get the data. Pinhole is there. Okay, grating is there. CCD detector is there, and all this integrated with software from the laser input to this. So you can collect the data. You can have this wave number versus frequencies. But one must know while using the Raman analysis how suitable the Raman is for this. So chemical suitability one must understand for the Raman analysis. So typically the most molecules with covalent bonds are Raman active. However, the nature and intensity of their signal can vary. And now the classical part of this Raman is for IR there are some limitations, but for Raman, yes, definitely there are no limitations as far as samples are concerned. In IR, if you look, if there is a huge water or moisture in the sample, so there will be a broad stretch of water, which will be prominently shown by the IR. So that's the problem with IR. But in Raman, water does not interfere in this Raman spectra, and that's the beautiful part of the Raman, wherein you can neglect the presence of the water and you can vomit out the interference of the water. Okay. So now highly fluorescent material that cannot be used or analyzed with Raman. That is limitation for this suitability. At actual Raman spectrometer as a PAT tool will look like this. So that's this the purpose. Purposely, I kept this slide. I'm showing you the slide. When such tools are used as a online PAT tools, so this is the scenario. Now, similarly for UV visible, I'm pretty sure that UV visible spectrophotometers are well known to pharma academics as well as the industries. Also, industry nowadays no one is using UV, but yes, academicians like us, yes, we are relying upon the UV. So we know the working range 190 to 900 nanometers, and it gives you the absorbance. Yes, pi electrons, double bonds, triple bonds, trines, dyes can be easily quantifiable by using this technology. Yes, UV visible spectrophotometer. Offline instrument will look like as previous slides gives you the idea about the offline instrumentation. This is something like instrumentation. Light source is there, entrance slit, dispersive element is there, then exit slit is there, sample, and then detector. Monochromator system is there. So entrance and exit slit. This is for the student purpose. So there are detectors, photomultiplier tubes are there, Indian gallium arsenide photodiodes are there, lead sulfides are there. So UV, this is working mechanism. UV is very common tool for UG as well as PG, so I'm not going much into much detail of working of this UV. Yes, but it works on Lambert's and Beer's law. A is equal to A into B into C. Okay, A is more, small a is molar absorptivity, B is path length, C is concentration. It's on the tip of tongue for everyone. So Lambert's and Beer's law is there. Okay, the absorbance is directly proportional to the path length as well as concentration. So this is the principle. But at actual, when UV is used as a PAT tool, now it is different instrument. You can see the probe, you can see the displays, you can see the inline mechanism by which you can use the probe. Okay. Now HPLC. HPLC is mostly most acceptable tool in the industry because of its sensitivity, accuracy, adaptability, automation, suitability, and applications. So it's a highly used industrial tool by the industrial tool. HPLC is very well acceptable by the industry because it has the ability to resolve very similar chemical components. That's why it is high performance. Correct? It's a high performance. So now HPLC, if you look into the working of the HPLC, everyone knows it. Just for the sake of students, this is the instrumentation and working of the HPLC. But when it acts as a PAT tool, now scene is different. You can see HPLC column, it goes into detector system, sensors are there, and based on this sensing system, you can collect the product or you can throw it out. So sample A, B, C, depending upon its collection and mechanism, it is kind of prepared to HPLC and it is automated as a tool. So Andres Hauser has developed this. Okay, process analytical tool references are there. So when HPLC works as a PAT tool, now see the changes. Now mass spectrometry, this is instrument which is available in my department, QTRAP. I'm lucky enough to have this instrumentation. I'm thankful to DST also, which gave me good support. So mass spec, you know, it was launched in 1906. J.J. Thomson, he got the Nobel Prize also. It determines the M by Z, mass to charge ratio, molecular weight. In layman language, you can say molecular weight, but it's a M by Z, mass to charge ratio. Okay, so in mass spectrometry, there are different instrumentations. Orbital mass spectrometry is there, QTRAP technology is there, triple quadruple, single quadruple tools are there. Okay, tandem spectroscopy was, mass spectrometry was launched in 1968. So what is the instrumentation? How it looks like? It's a sample introduction is there. Rest all is in vacuum. Source is there. It converts that into droplets, fine gaseous phase. Then analyzer is there. Ion sorting is there. Q1, Q2, Q3. If you have triple quad, Q3. If you have single quad, Q1, quadrupole. Rather, I should say quadrupole. Q1, Q2, Q3, or else Q1. You have ion detection system, then data system integration, and then mass spec. So sample inlet is there, ion sources are there. So ESI, electrospray ionization, is most common source of ionization which is used in LCMS. ESI, APCI is also there, but ESI 99% molecules can be fragmented by using this ion source ESI. Okay, analyzers are there. So now working, you know it, how mass works. Just go through instrumental methods of analysis by Willard, you will get the idea. Basically, it is samples are injected into vacuum, ionized. Ions, they get charged, they get deflected, and based on deflection and M by Z, there is a segregation of the ions, and that can be detected by the electron gain detectors. Okay, so typical mass spectra will look like this. M by Z, it is from 50 to 600. This is sample chromatogram, ion chromatogram from my lab only, along with, so it is M by Z versus intensity, abundance in CPS. So you can see the various fragments. So depending upon your material, so M by Z, it has been resolved by the mass. But when it comes to PAT as a mass, so see, I'm sure figure is not that much visible, but resolution was not up to mark. See, reaction system is there. If you are synthesizing something and if you connect that to the mass, so it will get coupled with mass and it will go into mass and it will identify that. So in this reaction system, you can measure the reactants, you can measure the intermediates, you can measure the products, you can measure the solvents. Okay, it's a gaseous phase, no? So you can couple this inlet as an inlet to the mass spec. So mass spectrometer can be used as a PAT tool also. You can see the reference also. Now, the technologies which are little bit unknown to us, focused beam reflectance measurement, focused beam reflectance measurement. So Metler is the Metler Toledo is the company which is leading company in this FBRM. FBRM is patent, basically term is patented by Metler. It is non-destructive tool, and basically it is typically used for particle size. So we are more interested to know the particle size. Those who are working in nano or nanoparticle systems, nanonizing, okay, nanomaterials. So particle size is very very important. So if that particle size, if you want to measure it online, then FBRM techniques is widely preferred. It is based on backward light scattering. You can get the image. You can see some of the working parameters in this slide. See the parameters. So you can see the workings also. So from laser to backbone. So focal position is there, and you have sample systems. You can backward imaging, and you can collect the data. There will be some threshold, and you can get this. See, every instrument cannot. It is not possible to cover in the cover the mechanism in the presentation, but just to give you the glimpses. Okay. So at real time, focus beam reflectance measurement tool will look like this. Okay. Now, NMR, all of you, NMR is it works in 4 to 900 megahertz. Basically, it is C13 1H NMR, number of hydrogens, number of carbons you can quantitate. Okay, based on it is a structural requirement. So Nobel Prize again, it was awarded for this NMR invention in 1946. Felix Bloch, they invented that, and Edward Purcell. You can see the eminent personalities here. It is based on resonance technique. So this is an instrument. You can have magnet systems. You can have NMR console and workstation. So helium is there, nitrogen is there. Field sweep and frequency sweep. Both the equipments are available. You can have field sweep. You can have frequency sweep. So now you can have megahertz to gigahertz equipments right now. So working is there. You can see the working of the NMR. So NMR graph, as simply acetic acid, if you can see the chemical shift in ppm. This is the important slide which I would like to show you. When it is in a reaction mixture, see, this is the actual photograph. This probe, it is going towards the NMR. So NMR platform can be coupled with reaction, and you can monitor the reaction online. Online monitoring reaction is quite possible. Now see the acoustic resonance spectrometer. ARS is another technology which is very important. Acoustic resonance spectrometer. It is based on, it is again non-invasive, non-destructive technique. So it has an application in chemical reaction checking, blending, polarization, and fluidized bed granulation. ARS is again a very classical PAT tool which analyzes sound generated using in pharmaceutical processes. Now see, you transmit the sound waves, it goes through sample, it is received, piezo discs are there at the time, and it is a sample, it through passes. See, this is the classical way of explaining white noise excitation. It goes to sample, it generates some spectra and calculate it. You get the spectra and interpret the data. At actual, how it looks like, actual photographs, ARS as a PAT tool. See, you have to fill the sample here, piezo discs are there, you have to supply the sound waves, it will go through it. It is basically based on vibrations. It is based on vibration. Every chemical has a signature, vibration signatures, and that signature is recorded in terms of your spectra. Now, what are the PAT applications? So, identification of raw material, crystallization, drying, milling, blending, drug product content uniformity. So, this is one of the classical graph which will help you. If you want to analyze raw material dispensing, NIR, Raman tool is there. For reaction monitoring, mid-IR, NIR, UV visible is there. For crystallization, mid-IR, Raman is there. For drying, wet granulation, NIR. So see, the classical way of using the equipment for the PAT. So it's NIR which is having a major role. So identification of raw material, handheld Raman spectrometer is there. It is again for by Brooker. You can see a person is directly checking the material at the site itself. Okay. So mid-IR can be used for this. Then crystallization. So ATR based FTIR probe is there, FBRM is there, temperature probe is there. So you can measure the crystal size, exact crystal size also. So what are the crystals? Because at the last crystallization, you have to add some seed crystals. So size of the crystals which is generated in the bulk, which has to be monitored. The FBRM will help you there. See, NIR, MIR is there. FBRM measures the change in particle size and particle count, and formed crystals are investigated using particle vision microscope. PVM also is also there. A figure is there in my presentation. I can show you there. So online drying, online monitoring, improved cycle time, reduced analytical work. So in drying, solvent monitoring is done by MS. Even the final product which is generated can be monitored by the mass. Okay. Now for the billing, see, particle video monitoring. So PVM, FBRM is there. If you are trying to mill some product, it's a unit process. You all know pharmaceutics. So in milling process, online particle size analyzer based on reflection, laser diffraction, and if you monitor the milling quality of the material, so you can improve the efficiency of the milling operations. So this is the classical way samples are sent through Venturi, and it goes to process line, and in Venturi eductor, PVM, FBRM tools are there. So particle video monitoring is there, uh, and you can have reflectance measurement. Focus beam reflectance measurement is also there. So now in blending also, again, see, for blending, you can achieve the uniform blending. How to know it? By online pack tools, you can achieve the blending, and you can assure whether my blending is proper or not. Because see, contain uniformity is typically based on blending. Blending is very important unit operation as far as pharmaceutics is concerned, correct? So in that case, near IR UV can help you there. One more tool which I would like to tell you about, laser induced fluorescence. So this figure is there, laser induced fluorescence is there. So laser beam is there, sheet opticals is there, fluorescence, it is generated here, it is collected, CCD cameras are there, and you can have display. So material, if it is there, so through the laser beam, so you can have this laser induced fluorescence, and blending can be verified by using this NIR, laser induced fluorescence, or even UV probes. Now, the next part of this my presentation, up till now we were more towards the PAT tools. What are the analytical techniques or tools, inline, online, offline tools which are used by PAT in PAT framework? But I would like to tell you about some of the universal analytical tools and its validation glimpses. Because method validation we do have some insight, but for instrument validation, IQ, OQ, PQ, that becomes essential. And there are some tools for which data is almost unavailable. It's very difficult to track the data. If you refer my presentation, if you try to collect data, it is quite challenging to get some crucial data of instrument validation as far as universal analytical tools in PAT are concerned. So for IQ and OQ, I'm not going to talk about IQ, OQ, but yes, I'm going to talk about PQ, performance qualification. So if you talk about first part, see, near IR calibration. So if you are using near IR calibration and near IR for as a PAT tool, so one must know how to calibrate the near IR before using. And see, in pharmaceuticals, whatever equipments we use, we need a regular calibration of that equipment and performance checks. Yearly, one has to have a calibration. So these test process, protocols I have specifically written for the students, those who are actively involved in this session. So you can have prepare samples, load the samples, measure the spectra, analyze the data, compare the peaks, and evaluate accuracy. While I'm explaining the acceptance criteria, you can read the test procedures also. So wavelength accuracy should be within plus minus 1 nanometer. Spectral resolution, okay, at full width at half height, it should be less than 10 nanometer. Photometric accuracy plus minus 5% of the true absorbance. Reproducibility and precision less than 2% RSD. Baseline stability drift should be less than 0.001 absorbance units. Linearity, linear regression coefficient, R square, should be greater than 0.99, and signal to noise ratio should be at least 100 as to 1. So you can actually verify the performance of the near IR before using it, even during the use. Okay. So This is for validation of near IR. Now, for Raman, if you want to verify the performance of the Raman, performance qualification. So see, in Raman, there are four typical components which are mandatory to calibrate, recalibration, even for new equipment. First instance, X axis calibration, Y axis calibration, laser calibration, and calibration or validation of accessories, CVA. CVA is also needed. So classically, if you want to calibrate x-axis of the Raman spectrometer, so toluene acid nitrile is needed, and test procedure is there, and acceptance criteria is plus minus 2 per centimeter is the acceptance criteria, plus minus 2.5 as established by ASTM. So you can see for ASTM standard, the acceptance criteria is slightly loose as compared to regular criteria. For y-axis calibration, again, metron Raman, you can use NIST traceable standards. Laser calibration, you can do it if you are operating at 785 nanometer. Beyond that, you don't need any calibration standards. But for 785, if you look into, so the wavelength accuracy plus minus 0.1 nanometer, laser wavelength should remain stable within plus minus 0.1 nanometer. And for CVA, polystyrene is the standard which is used. For the student purpose specifically, I have highlighted some of the key points in this instrument validation. So there might be some questions from the interview point of view, wherein what standards are used. So purposely I wrote there. So for Raman, you can use polystyrene, you can have these solutions, and you can recalibrate your instrument. Now, for UV, basically, solutions for checking the absorbance accuracy, potassium dichromate is used as a solution. So, stray radiations, absorbance stability, wavelength accuracy, so acceptance criteria are there. You can see the measured absorbance value should be within plus minus 0.2% of expected values. Percentage stray radiations you can minimize, you can measure. It should be less than 0.1%. So absorbance stability, baseline stability, noise at zero level, wavelength accuracy. So all these parameters, absorbance accuracy, stray radius, it has to be ensured. So while using the typical tool as a PAT tool, one has to have a calibrated equipment, calibrated equipments. Now, as far as HPLC is concerned, HPLC has more components. So my two slides are covering these tests and acceptance criteria. So I'll be spending some time on this slide because HPLC is a very vastly used tool. Although online, offline doesn't matter. What matters is calibration of the equipment, performance of the equipment. So PDA, UV visible detector, PDA or variable wavelength detector. So you have to check the wavelength accuracy. Wavelength accuracy has to be checked. Uracil, holmium oxide, caffeine can be used. It is written over there in test protocols. If you want to check the performance of the pump, flow rate accuracy, 1 ml per minute, set the flow rate, take the flask and fill it, volumetric flask, calibrated volumetric flask. Within 10 minutes, it should give you the 10 ml. Okay, acceptable criteria, 1 ml plus minus 0.05 ml per minute. This is the acceptance criteria. Compositional accuracy, you have to use gradient, 0.1% acetone in water as well as water. You have to set the gradient. Those who are effective users of the HPLC, they can understand very well. Set the gradient in a steep manner, set the flow rate at 2 ml per minute and check. The calibration ranges should match with the desired calibration and R square should be more than 0.99. Auto sampler has to be calibrated, performance checks should be there. So repeatability of the peak areas, RSD values are written there. The control samples has to be run. Wash injections should be there, again run every 2 samples injections. You have to in between that you have to wash the injection system. So auto sampler has to be calibrated. Now next, column 1. If you talk about the column 1, yes, it is again integral part and heart of the HPLC because temperature affects the separation, affects the chromatography. You know it very well. So for that, column 1 should be calibrated on, acceptance limit is there. Set the column temperature. Use a typical flexible thermocouple. NIST traceable digital thermometer is there. You can use that also, or else you can have thermocouple flexible and check whether if you set for 40 degrees centigrade, it should be within plus minus 0.5 degrees centigrade. Huh? So limit is it should be variation should be less than at different locations less than 1 degree centigrade, plus minus 1 degree centigrade. Columns, yes, definitely. Plate count. Column for column, I know people are very well aware about the theoretical plates, number of theoretical plates, height equivalent to theoretical plates, resolution, peak asymmetry, everything you know it. Annotations, go to annotations, you will get the data. So two peaks, what is the resolution, how they are different from each other, check it. Plate count should be more than 2000 plates per meter. That is the standard requirement. Okay, resolution should be greater than 1.5 between critical peaks. Say suppose you are separating the curcumin, desmethoxycurcumin, bis desmethoxycurcumin. These peaks are very close. I am a chromatographer since last 20 years. I am doing it. So it's very difficult, challenging to segregate those peaks which are chemically very similar. So in that case, resolution matters a lot. So it should be more than 1.5. These kind of parameters you have to test for your columns before using the columns for actual online monitoring. Wavelength qualification is there. Holmium oxide is the standard in 10% per caloric acid. NIST traceable standard is that. Calibrate for caffeine is also secondary standard. So you have to calibrate the wavelength detector wavelength qualification by holmium as well as caffeine. Okay, so wavelength accuracy precision is there. So percent RSG values are also written over there. So this is for HPLC tool. So for NMR, it's a huge protocol. I'm not going to read that. I'm just showing you. Even for NMR, if you are using NMR as a tool, yes, magnet has to be calibrated. So again, NMR is having magnet, NMR probes. So for NMR probe, there is a huge, basically procedural part. Those who are actively involved in the NMR operations, NMR operating, they can understand very well. So you have to check the magnetic field homogeneity. So acceptance criteria is zero point 0.1 ppm or better for high field magnets. For NMR probe, plus minus zero point 0.1 hertz is the acceptance level. So you have to prepare lock procedure is there. I will move the slide to next. See the protocol. Again, radio frequency coils has to be checked for its performance. So sensitivity test is there for radio frequency coils, because if you are trying to resonate molecule, it is based on coils only, correct? So if you understand, if you recall the theory from instrumental methods of analysis, so sensitivity test has to be done. The signal intensity should be uniform within the sample volume with variation typically less than 5%. Okay. So for NMR calibration, these are the three typical protocols which are mandatory. Now signal to noise ratio, again that is important part. Acquire NMR spectrum with standards. Signal quality, check the quality, repeat the test. Variation in the test, just see, it should be less than 5%. Now these are the references for this my presentation. Now this is little bit about my department. Recently I got a Center for DMPK Studies. It has been approved by the then Honorable Vice Chancellor Dr. Pramod Yevle sir. I got this facility well supported by the DST, National Facility for Bioanalysis, and I'm lucky enough to I do have GCMS, LCMS, HPLC, high throughput equipment like absorbance, fluorescence, phosphorescence, plate reader, that is there with me. And through this center, national facility, we are offering services to students, academicians, as well as industries. So we do have currently good industrial projects also. We do offer services in visible spectrophotometry, HPLC, LCMS, GCMS, ELISA plate reader. GCMS facilities are little bit restricted for outside, but other than GCMS, we are currently offering every facilities. We are more into antioxidant assays, isolation of phytochemicals, purification of phytochemicals, and we do offer contractual research services also. So this is my team, my students, my beloved students, of some old batch students, my new batch students. This is a typical view from my laboratory, and this is the conclusion part or end part of my presentation. So thank you very much. I am thankful to KT Patil team, Patil sir, as well as Joshi sir, Madam, Biradar madam, and entire team for giving me the opportunity to present my views in this pack. Thank you. Thank you very much.
Thank you.
Thank you.
Thank you,
So
sir.
now, if there are any questions, so please let me know. I will try my best to answer your questions.
Yes, sir. Surely, sir. Sir, the session was very informative, and while looking into the chat box, our participants have asked some questions. So, shall we start the question answer session, sir?
Ha. Yes, sir. Yes, ma'am. Your voice is little bit disturb.
Okay.
Okay. Just ask me. I will tell. First question.
One of the participants asking about what are the applications of AI in PAT, sir?
Ha. Acoustic resonance spectroscopy is vibrational spectroscopy basically, and it helps you to identify the contents also. ARS can be used to content determination, particle size determination, even packaging materials also. If any packaging material is hollow in the nature, so online ARS tool can help you there to see the empty spaces of the packaging material also. So ARS is one technique, but how to use it, it depends upon us. So there are various applications as far as ARS is concerned. Next,
So next, how does PAT handles variation in raw material or any process condition?
huh. In PAT is an online tool. Basically, it actually monitors the real time scenario. So I told you about the process control tools of the PAT also. If something is not right with the starting material, so PAT control tool will let you know your product is not into specification. It will not allow that process to go forward, and that is the classical way to monitor the process in real time scene. So if there is, say suppose you have paracetamol and your specification limit is 99.2% paracetamol should be there as far as API is concerned. And if it is less than 99%, and if you have PAT tool along with that, and if it is showing the assay is only 98.5, so there is deviation by 0.75, it will trigger the impact and it will stop the process immediately. It will let you know, it will give the indications. No, your assay is not in specification limit, stop the process. It will not allow the process to go move forward. Next.
Okay. Next, are there any concerns about data security and privacy in the PAT system?
Constraints about data secrecy.
Concern about data security and privacy in PAT systems, sir.
Data security, yes, correct. That is another challenge. Basically, right now, the data which is collected, we collect offline data. We try to collect the data in hard copies and in some soft copies, and we try to store the data. But for online pack tools, data collection and data storage is quite challenge. Right now also it is challenge. But now we do have 21 CFR compliant systems which collects the data, real-time data, and which collects that data for us. PAT services offering companies are now offering the softwares which can collect the data and which can store the data for you. And as I say, PAT is a segregated tool. If I apply my PAT framework for my industry, definitely my data will be stored in my custody only. So no need to worry about the data. But only thing is you must have a very secure software which can collect every data points. Next,
Can we apply PAT in green chemistry, sir?
can we apply PAT in
Green chemistry.
green chemistry? Yes, definitely. PAT framework, PAT tool is for real-time monitoring and correction factors. You can have green synthesis, you can have any type of synthesis, you can have formulations. It will help you to control the parameters right now in the process itself. That is the beautiful part. Green synthesis, no issue at all, because critical variables will be similar in green synthesis also. Only thing is you will be having processes which are modified processes, but the crucial parameters will remain same. So PAT can be easily applicable to green chemistry. Next,
So, thank you, sir. All questions. You have resolved all the
thank you.
questions. So, the questions are over, sir. So now on the behalf of Other Shikshan Prasark Mandal, KT Patil College of Pharmacy, I would like to present certificate of appreciation to Dr. Sachin Bhusari sir. Please accept sir. Please accept sir. So now it's time to present a vote of thanks. On a behalf of other Shiksha Prasarak Mandal and on the behalf of all the participants, I am very much glad to present a vote of thanks for first session of this one-day seminar. My heart fills with lots of gratitude and respect for our today's knowledgeable and eminent personality and the speaker, Dr. Sachin Busare sir, not only for sparing their valuable time and knowledge with us, but also for enlightening us with their admirable talk on such a crucial subject. Also for clearing our concept and enhancing our understanding pertaining to the same subject. So thank you, sir, for such an in-depth session. And I also uh, we will always very grateful for your kind cooperation for the success of this seminar. So thank you, sir. Thank you so much, sir. Lastly, I would like to thank all the participants for your overwhelming response. Thank you. Thank you, one and all.
Thank you.
Thank you, sir. Now there are some instructions for participants.
Thank you, sir, for giving your
Thank you.
valuable time to share very, and it is very much useful for the student as well as the staff because it is a new topic. So thank you very much. Thank you once again.
I tried my best, ma'am. Thank you very much. Special thanks to Birazda ma'am. Thank you very much for giving me the opportunity. Joshi sir and Birazda ma'am. Thank you so much. Special thanks to both of
Yeah.
them. And thanks to audience also for their
Thank you, sir.
patience listening. Thank you.
Thank you, sir.
Yeah.
Now there are some
Every year will
initial.
give you new topic for the
Thank you.
yes.
May I leave, ma'am?
Yes, sir. Now there are some instructions for participants. It is the first session was over. There will be a break for 1 hour, and the second session will start sharp at 2 p.m. So have a lunch and join again at 2 p.m. Secondly, for the issue of e-certificate, it is compulsory to fill the feedback form. The feedback form link will be shared in the second section. Please do attend the second session. So now with a due respect, I would like to conclude the first session. Thank you one and all.